In chapter 7, we're going to be going over the cellular basis of inheritance. So, as we move through this chapter, recall that a gamete, the term gamete, is the same thing as a sex cell, and that it has half the total amount of DNA as the parent cell. And remember also that another word for gamete is germ cell or a sperm and an egg. Also remember that mitosis makes body cells, but meiosis, and some teachers say meiosis, but I prefer meiosis. Remember that meiosis makes sex cells. So, and also remember that in the last chapter, we talked about binary fission, which is just the splitting of a cell, which basically without it's a type of reproduction where it's just split. It makes a clone, it makes a copy, and it results in just as much genetic material in the parent as the offspring. But in this chapter with meiosis, we are going to talk about how meiosis results in half as much genetic material in the offspring as the parent cell. From there, the two parents, once the daughter cells have half the number of DNA, the two daughter cells are going to basically combine an egg and a sperm to mix and make a baby. A child that has the DNA that's different. It's a new mix from either of the parents. So this process of making separate sperm and eggs is called meiosis. So there are some single-celled organisms, and I'm just going to throw out bacteria as an option there. There's a few single-celled organisms and a few multicellular that make identical clones of themselves through meiotic division. But for the most part, while mitosis makes clones, meiosis makes unique individuals that are different from their parents. And sexual reproduction is the production of parents of haploid cells, so having half the amount of DNA, and then the fusion of two haploid cells from each parent to form this unique diploid cells. So why would any organism want to sexually reproduce? It's a lot of extra work. There's exposure to illness. There's the needing of a mate. So in other words, you have to be in close proximity. That's not a big deal for some organisms, but some that have a large range that they live in, that can be problematic. Why do any creatures do that? They do that because of diversity and increasing variation. So every time there's a new mixing of genes, now instead of having clones, you have some variety. So sexual reproduction is the preferred method of reproduction when environments are favorable because it allows for diversity. And then when there's threats facing the population, hopefully there's going to be enough diversity that at least some of the organisms in an existing population will survive. While some might die, some might have some really good genes in there that are able to survive whatever new illness or plague or environmental changes come along. So like if a new virus comes, hopefully somebody's got the right genes to survive. Okay, I want to talk about this picture in your book. So there's three main categories of life cycles in multicellular organisms. Humans have a diploid dominant life cycle where we generally have diploid cells, okay? And this picture here shows that there's some organisms like fungi have a haploid dominant life cycle, and then there's actually some plants and algae that actually switch between having a haploid and a diploid life cycle called the alternation of generations. And if an organism is going through alternation of generations, you can tell if it's haploid or diploid by basically counting the number of chromosomes. And we'll look at some karyotypes, they're called, that you can count chromosomes at the end of this. So, alternation of generations has a diploid and a haploid stage. So, like in these ferns, during the spore carrying part, it's a diploid, meaning it has both copies, and so it's making a spore. And then during it actually has a phase where it can have eggs and sperm, and that's a haploid generation part. So, in animals, sexually reproducing adults form haploid gametes from diploid germ cells. So that's back up here. In animals like humans, we have a haploid point in time, but the majority of our body cells are diploid. And try to remember that haploid means that the organism only has one copy of the gene. So that is when we're in our sperm and egg. Like when the sperm are made, or eggs in the female, those are their haploid, but the majority of our body cells, called somatic cells, are actually diploid in humans. So, what else do you need to know here? Fertilization between gametes, so the joining of this egg and sperm, makes a zygote, which is a fertilized egg, and it's now diploid. In meiosis, a diploid cell has two copies for each trait, and it will give rise to a haploid cell. So you can get one set of genes from mom and one set of genes from dad. To do this, the cell divides twice. So let me see if we can get a picture of this. In order, I think I want to go back. I'm going to go back because I think that would be a better picture. Okay. To get this haploid phase, the cell actually has to divide twice, so it has the amount, the right amount, which is half the number of cells in an egg or a sperm, and that makes 4 haploid daughter cells. And this process is called meiosis. This is what we're going to talk about. And then, in addition to fertilization, sexual reproduction includes a nuclear division that reduces the number of chromosome sets. So we'll talk about that in the next section. Okay, we'll move to this picture. So at the beginning of sexual reproduction, which is fertilization, there's a union of 2 cells from 2 individual organisms. So a mom and a dad, and then those haploid cells, haploid means half, so those 2 haploid cells have half the number of chromosomes. Then when they're joined together, an egg and a sperm, it makes one individual that now is diploid, or has the full number of cells. Then try to keep in mind that our body cells are called somatic cells. Okay, try and keep that in mind. There are cells that they do make copies of themselves, but they're not for reproduction like sperm or an egg. Also, when we go ahead and move into karyotypes, many of you know this already, but human females have 2 X chromosomes on their 23rd or last pair, and males have an X and a Y. There's a few other combinations out there, but those are going to result in various disorders, and we will actually talk about a few of those as well. So the first phase of meiosis is called Well, meiosis is preceded by a phase called interphase, and it's largely the same thing as interphase that we had in mitosis. There's a G1 phase where the cells grow, there's an S phase where the DNA is replicated, and then there's a G phase where just final preparations are made. Things are cross-checked and that kind of thing to prepare for meiosis 1. So in meiosis 1, homologous chromosomes come really close together and pair with each other, and that's called the synapse. There's, and this is similar to mitosis because both in mitosis and meiosis, there's homologous chromosomes. So in your cells, chromosomes exist in pairs. You only get one copy from mom and one copy from dad, and these are called homologous chromosomes. So just like what you're looking at here, these can be homologous chromosomes. So these don't code for the exact same gene, but they code for similar genes. So what I mean by that is that this region of A on this one and this region of A on the other side might both be coding for something like hair color. So they both regions are coding for hair color, but obviously you might be getting a different hair color from mom than you did from dad. In fact, it's highly likely that you're getting a different hair color from mom and from dad. So they're homologous, meaning they're coding for the same types of things in the same place, but it's not identical like identical twins. So sister chromatids are the sister chromatids, like this side and this side of the same chromosome. These two sides are going to be identical to each other. But then meiosis is going to remix things so that these two sides are not the same as we move along. Meiosis makes the cells different from each other by having something called crossing over, where there's a remixing of genes. And you can see how these guys mix up genes here. It's important to note that crossing over does not create new versions of the genes. It just makes new combinations. So basically it just recombined, like crazy sock day, like switching socks or clothes or shirts or whatever. It just is mixing up some of the genes so that now this side is not identical to this side anymore. So they're different from each other now. And this side of the red X is not the same as this side. So that's called crossing over, and it just makes new combinations of genes. So in meiosis, independent assortment does occur. This is called independent assortment, which makes unique combinations of genes, and that's important to remember. Independent of assortments of genes happens in metaphase 1, and it's sort of like shuffling a deck of cards, and the same cards are all there, but now just in a totally different order, and you don't know what you're going to get. Humans have 23 chromosomes in their gametes or sex cells in the end. Your body cells, your somatic cells, remember they have 46, but your sex cells only have 23. So when you have, you combine that egg and the sperm, then you have a complete set of 46. Okay. So this just further shows like chromosome arrangements here. I wanted to go over, let me see if it is here. Yes. Okay, without flipping. Okay, I wanted to go over some of the steps of this this image here. So here you have these homologous pairs of chromosomes, and they are held together at what's called the chiasmata. And then microtubules end up attaching to like their middle portion and start to pull them apart. Okay, so these sister chromatids, they remain with their sister, but they are pulled together from their homologous pair. okay? But they're still attached to their main one that they came in with. And then during meiosis II, then these sister chromatids are split apart to make the final division where you only get half as many chromosomes. Okay, so this chart is here too. So this is another really important one. So if you look at meiosis and mitosis, and if you were going to compare those two, notice that meiosis right here, meiosis has two divisions. So meiosis I and meiosis II. Whereas mitosis only has one division, one big long division. Also notice that the end of meiosis II looks a whole lot like mitosis division. Like if you just did this end here and then compared it to this division here of mitosis, these two look really similar right here. And it does. It just is in meiosis, it's happening twice, here and here. And in mitosis, it's just happening down here. What doesn't happen in mitosis is all of like what happens in meiosis I, okay? Because there's only one division here. So I'm going to walk you through this. In meiosis, you get 4 cells in the end that are haploid or half, whereas in mitosis, you get 2 cells in the end, and they're diploid, diploid, which means they have twice the number of chromosomes for your body cells. So this is body cells, this is sex cells. So in interphase right here, you have the G1, the S, the G2 stages occur, which is mostly like replication and getting ready for the division. And then you have prophase 1. This is where the nucleus dissolves. And then as you move through, you have metaphase 1, where the chromosomes line up. I always think met in the middle, metaphase for meeting up in the middle. And then you have the anaphase, where chromosomes are split in half. And then in telophase or telophase, you have the cleavage furrow, or you can have, if it's a plant, a cell plate that forms. And then you go into meiosis II, where roughly the same thing happens, but in the end, in anaphase 2, the sister chromatids are ripped apart or split apart, which gives you the 4 haploid daughter cells in the end. And here's a little chart from your book that goes over each of those again. So just make sure that you look at that. Okay, we're going to go to the next section. Sometimes there's mistakes. Sometimes there's mistakes in meiosis, or errors in meiosis. Sometimes sex cells don't split correctly. They either split too many times, which results in too few chromosomes, or they split too few times and you can have extra chromosomes. So these mistakes can cause different disorders such as Klinefelter syndrome or Turner syndrome in females, Klinefelter's in males. And this is a phenomenon that is called non-disjunction. So what you're looking at here is actually the karyotype, or all the chromosomes of a typical human. And you can see that there are 2 homologous pairs at each chromosome. When you initially do this, these are all just like floating around, and you actually have to pair them up like a puzzle. And you can see on the 23rd pair, you have 2 chromosomes that are the same size, which indicates that is a female. If this was a male, you would have 1 tall X and a very, very small, like maybe this smaller, maybe even a little smaller Y chromosome, and then that would indicate that that is a male. So what are some of these? So I have actually maybe a better PowerPoint from Zahur Rami, and they had these mistakes in errors that can be made, and I liked these pictures better. So I'm sorry, I'm going over to this presentation instead. So one of them is called non-disjunction, and non-disjunction means if I have cells normally, they're going to split, and then you should have 4 haploid daughter cells. This is a very easy picture to see. In non-disjunction, if this didn't split properly here, so the non-disjunction occurred at this point in meiosis I, then all of the chromosomes are just going to go to one cell, and this one isn't going to get any. And then as it splits again, you're going to have twice the number of chromosomes. Remember, this is the correct one. You want to have 1 chromosome, but instead you have 2. Another way that non-disjunction can occur is that it splits correctly in meiosis I, but in mitosis or meiosis II, it then just all goes into one cell. Now this side split correctly in this picture, but this side did not. And so that's how you can get non-disjunction, meaning it did not split correctly. So aneuploidy, which is it can be harmful because the dosage of the gene can either be too high or too low. One example is trisomy. On the 21st pair, if trisomy happens on the 21st pair, then you can get Down syndrome. And there's an extra chromosome on the 21st pair in every single cell of your body. So your book calls this trisomy. This PowerPoint presentation calls it polysomy. Monosomy is only having 1 copy of a particular chromosome when you needed 2. That can make Turner syndrome. In females, they're missing one of their X chromosomes, so they have 1 X, but they don't have their second X from their dad. And like in no, that's a good example of monosomy. So there's different errors that can be made, like something can get deleted where sections are missing, things can get duplicated. There can be inversions where parts of the chromosomes get twisted upside down, or translocations where full sections can move over into other sections. Like here's a translocation where a piece of this chromosome just joined on to this other chromosome and did this. That's like a translocations. So you can find these errors by doing a karyotype, and a karyotype is where all the genes are mixed up, and you just need to make sure that there's kind of somewhat matching pairs at each chromosome level until you get to the last one. And you can see here's a bigger X and a smaller one next to it. That means it's a Y. So that's obviously a male, like we talked about earlier. So you can actually have sometimes 3 chromosomes in a certain place. So it's possible to have trisomy on 13, 15, 18, 21, and 22 and survive. But it's usually fatal if it happens anywhere else. So on this next slide, look and see if it's a non-disjunction or if it's a chromosomal error, meaning one chromosome is too long or too short, or if it's non-disjunction. So if we go and look at this, okay, there's a matching pair, even though some are like kicking off to the side. Here's matching pairs. What's wrong with this one? 2, 2, 2, 2, 2. Whoa, there's a 3, right? There's 3 chromosomes right here. It should only be 2. So that's trisomy, and it's on the 18th pair. So that would be like a non-disjunction that happened there. So most DNA changes like this results in big changes for humans. But if humans have an extra X chromosomes, sometimes it can be inactivated. X inactivation is canceling one of the extra chromosomes. The canceled extra X chromosome is called a bar body, and the genes don't get expressed. And I want to show you, I want to go back to let's see if it's here. Okay, it is. Um, so here's a picture of a tortoiseshell cat. A female tortoiseshell cat has color on two different chromosomes on its body. In this case, there's coding for black on the X chromosome, and then there's coding for brown on the other X chromosome. So, depending on which body part um, the chromosome was working on depends on what color gets expressed there. So one chromosome, when it's making over here on its thigh, it's like, oh, okay, well, this chromosome that was working over here just happened to have brown, so it pushes out the brown hair color. But down here on its tail, it had a chromosome for black, so it just gave it a black tail. So basically it ends up making this patchwork of color that gets expressed differently in each area. So some errors in the sex chromosomes have different names. For example, for males, you can have an X, and a Y. And this is called Klinefelter syndrome, and it corresponds to males. Some of the things that they can have is changes in their testes or breast development, reduced body hair, and it's because that extra X chromosome undergoes inactivation to compensate for the extra genes that are there. So these are called polyploid when you have more chromosomes that you need. In the animal kingdom, it's very rare, but you can have them with some flatworms, crustaceans, amphibians, fish, lizards. In the plant kingdom, it doesn't cause as many problems to have a polyploidy. So, and sometimes you can just have an organism that's a much bigger version of the plant. Here's another syndrome called Cree-de-Shaw syndrome, and it's missing part of the fifth chromosome. So the fifth chromosome is just missing a small part, but these individuals have when they're babies, it's characterized because their head is potentially a little smaller. They also have a high-pitched cry. In fact, in French, it means cry of the cat. And it can be fatal if the child survives till 1. Usually they'll have a regular life expectancy, which is great. But there can be some cognitive delays there. So that's everything that we needed to go over in this chapter. I hope you look it over yourself because we go through it very quickly. Good luck as you continue to study about meiosis.